Good morning, welcome everyone to for this session. So I'm really happy to see that the room is quasi full. So this session is, the title is a mechanistic approach of the varietal mixture effect on plant pathogens. So I'm very happy to co-chair this session with my colleagues from INRAE, Elisabeth FOURNIER and Jean-Benoit MOREL from Montpellier. So and in this session, we will talk about challenge and issues about, so in a broad sense, biodiversity within the field and especially cultivar mixtures. And our global question is, are cultivar mixtures in the world, is there a success story? So the first statement is that it works. I mean, mixtures, so I take example in several crops, reduce the disease, several diseases. So you have on this first introduction pictures, only brief summary of this example. So for instance, bread wheat varietal mixtures in France and in Denmark, so in broad sense in Europe. Also durum wheat land races, so we are a high level of diversity within the field in Tunisia. And also old example is barley mixtures during the previous, the last century in former DDR, used to control powdery mildew. It's a very documented example. And the last, most recent example is the eternal rice project in China. So I let Elisabeth present the different presentation.
Thank you. The objective of the session is to show that mixtures really work and to understand how they can better work in an evolutionary context. So in their paper published 5 years ago, thank you, Borg and collaborators reviewed the different biotic mechanisms that are in action in diversified plant covers and that allow to lower the burden of pathogen epidemics. So these mechanisms, some of them are involving the direct interaction between the pathogen and the plant, and they are of different kinds. And other mechanisms are acting, such as plant-plant interaction, also act without the presence of the pathogen, but they allow to modulate the immune response of the plant. So we will have 6 talks during this session, and all along these talks we will have highlights on these different mechanisms that are acting in diversified plant covers.
I'm very happy to introduce Elisabeth Fournier and Frédéric SUFFERT, who are both colleagues from INRAE. So one is working in Montpellier and the other one is working in Saclay, Paris. One is more working on rice, Elisabeth, and she's more of a pathologist and a geneticist, whereas Frédéric is working on wheat and is more of an epidemiologist and modeler. And they are going to tell you about their approach to see how much evolution is actually involved or not in cultivar mixtures. In particular, this work is part of a large French project now ongoing, which if you're interested in that topic, I recommend you follow up. This is the Combine project, which is gathering, I can't remember, but quite many labs in France on this very topic to see how, in particular, how pathogens evolve in diversified cultivar mixtures. Okay, so the floor is yours. Please.
So
thank you.
Wait. So the challenge we take is to question about if cultivar mixtures are a way to solve what we call the trade-off between efficacy of the mixtures to reduce the disease and sustainability goal, durability of resistance gene used in these mixtures. So I gave you the slide you already seen in the first introduction, and I let Elisabeth talk about the objective of the presentation.
Yes, so here we would like to highlight a recent body of knowledge on the multi-scale mechanisms that are in action in varietal mixtures and that can play a role on the control of plant diseases but that also can drive the evolution of the pathogen population regarding virulence or aggressiveness. So, as already highlighted in the session introduction, there are several mechanisms acting in varietal mixtures, and here we will focus on the mechanisms that are involving the interaction between the pathogen and the plant. So you have some mechanisms are like physical mechanisms such as dilution or barrier effect. We have also what we can call ecological mechanisms in action, but also evolutionary mechanism and especially disruptive selection. And these different mechanisms have different impacts, expected impacts. So, for example, the physical and ecological mechanisms are expected to play a role in the reduction of the size of the pathogen population, so reducing the severity of the disease, and they act at short term at the feet scale and year scale. But there are also these evolutionary impacts on some of the mechanisms, and this that means the change in the evolutionary trajectory of the pathogen population. And again, there are some mechanisms that will act at short term by switching the allelic frequencies or virulence frequencies within 1 year or one epidemic season, but also long-term evolutionary effects that will act on the among several years, and especially the disruption of the directional selection. So here we will give examples of these different kind of mechanisms. So Frédéric will talk about short-term epidemiological and evolutionary impact within wheat cultivar mixtures, and I will rather give you an highlights on this kind of mechanisms that are acting at the long term.
So, and the more general challenging question can be summarized here. So, in practice, can mixtures increase or maintain the durability of resistance in cereal compared to parameting the genes? And with new tools, I mean genomics, can we validate all those models that suggest in the past an effect on the evolution of aggressiveness and virulence in cereal pathogens? Which biological mechanism affects the population dynamics, and how to exploit and enhance this mechanism to gain in both efficacy and sustainability of resistance genes. So, and the case study would be cereal canopy, wheat and rice. So the first case study is about Septoria leaf blotch. So it's an important disease of wheat, and the most pregnant aspect of this disease is a coexistence of asexual and sexual reproduction. So you have asexual on the two pictures on the left and sexual reproduction on the wheat debris on the right. Another characteristic is a very huge size of population and consequently genetic diversity to the sexual reproduction. And what we can use to protect the crop and the system is resistant genes, so some are overcome. And for instance, STB16Q, with each the gene we have used in a study. So here a brief overview of the disease cycle. So the disease is the epidemic is initiated by wind-dispersed ascospores at the beginning of the season, and then over the course of the epidemic you have a dispersion of pachynodospore by rain, and at the end you have a sexual reproduction on the wheat debris, and the epidemic is recurrent like that. So if we put mixtures in this slide, so you can see in red is a susceptible variety, and in blue is a resistant. I use the term resistant, but in fact is I mean a variety that carry a resistant gene, but it can be overcome. And in blue you have also the ascospores that are considered to be virulent. That I mean able to infect the resistant cultivars. And then you have a mixtures, and during this over the course of the epidemics, several mechanisms that are well documented in the literature. You have an initial varietal filter effect that induce a dilution effect. It's only the virulent strain are able to infect the resistant, or the cultivars that carry the corresponding resistant gene. Then you have cross-contamination by pycnidiospore and barrier effect that is active due to this mechanism of disease dissemination. And then just after sexual reproduction, at the end of the epidemic, you have a change in the encounter probabilities between the different strains to cross, and different in the probability of encounter between virulent and avirulent strain. And of course, what we expect is that the mixtures has an effect and modifies this probability of encounter. And the question is, in a mixture system, what's happened about the proportion of virulent and avirulent strain from the beginning to the end of the epidemic, so the end of the epidemic after the sexual reproduction? And the main question is, do cultivar mixtures drive an evolution of the population, I mean proportion of virulent strain? And pictures is missing nothing. A student, PhD student, which defended the PhD student 1 year ago, did great work in field. So comparing different proportion of cultivars, mixtures, compared to pure stand. And the two cultivars were Apache, is a susceptible wheat cultivar, and Cellule is supposed to be resistant, but in fact it was it carry a resistant gene that was overcome recently. And the work was consist in collecting strain on the different microfield, on the different variety, and to estimate the frequency of virulent and avirulent strain. And this was done by individual phenotyping, so strain by strain phenotyping on seedlings. And the result is you have here a presentation of the proportion, the frequency of strain virulent is in colored, and avirulent is white on the cultivar cellule that carry the resistant gene, and on the susceptible cultivar that do not carry the resistant gene. So it's a change. So is a susceptible cultivar, you have an expression of the filter effect. So you find only a few proportion of virulent strain corresponding to the proportion of virulent strain in the landscape, and the filter effect here on the cultivar carrying the resistant gene, you have mostly virulent strain. So nothing happened at the early stage of the epidemic. But after that the dispersal occurred, what you observed is a beautiful expression of the barrier effect. So on the susceptible cultivar here present in the mixtures, you have an increase of the virulent frequency with the increase of the cultivars that carry the resistant gene in the mixtures. So it's an expression of a modification, if you see at the scale of the global canopy, an expression of the modification of the virulent proportion. And something interesting it was obtained is that after sexual reproduction, so on the debris coming from this canopy, so we have quite a similar structure. It means that the structure and modification by the mixtures is maintained after sexual reproduction. So that's a good news. It means that maybe we have a long-term effect of the mixtures for the subsequent epidemic. That's quite new result, but something interesting, and for that reason we focused on the biological mechanism, is that you can see on this bar that there is a decrease on the cultivars carrying the resistant gene, a decrease of the virulent frequency of a decrease of the frequency of the virulent strain, what we call a relation effect, and it can be analyzed that a kind of preventing the virulence fixing due to sexual reproduction. It's our biological hypothesis that sexual reproduction play a role and has a strong impact in the mixtures. Just to be brief and finished on this example on Septoria tritici, we identified new possible mechanisms in mixtures that it's caused by the change in the probability of sexual encounter between virulent and avirulent strain in the mixtures. And based on an aspect that I will not detail today, is that avirulent strain can reproduce sexually in plants that carry the corresponding resistant gene. So I mean, strains are able to reproduce and transmit the virulent or virulent allele, although they are not infecting the corresponding plant. And that can be due to different biological mechanisms that we are currently trying to focus on and work on. So a conclusion about this aspect is that smart wheat mixture management can be a way to reduce the transmission of virulence, and this is function of different parameters that are enough time to develop today. And the perspective is to combine different cultivars with complementary resistant gene, and of course to develop modeling aspect.
I guess I will be out of time. So to address...
Not yet.
Yeah, I have. Okay. To address the long-term evolutionary impact of cultivar mixtures, we had the great opportunity to work to develop a research project in a very particular system, rice growing system in China. So the idea here is to address the question whether mixtures and highly diversified mixture can disrupt the directional selection that is acting when you have only one cultivar plant that is deployed in the landscape. Ooh, okay, so this was a picture of the Yuanyang terraces. So you have to trust me. So the Yuanyang terraces are situated in the Yunnan province of China, south of China. And they are the landscape is constituted of small paddy fields that are built all along the mountain slopes. And so you have, and the particularity of this system is that you have a huge amount of different varieties that are cultivated in sympatry. So there are up to 100 rice land races that were recorded in the system, and some of them, such as Atsuche, are cultivated for centuries. And there is no chemical fertilization and no pesticides that are used in the system. And rice diseases are recorded, but there were no major sanitary crises that were reported. So to illustrate this, here you have a phylogenetic tree showing the diversity of rice accessions sampled in Yunnan in black compared to worldwide rice accession representative of the diversity of the different rice types. And so what you can see here is that most land races are indica land races, but there are also some japonica land races that are cultivated together with indica land races in the system. And the second important thing is that we really have land races, that means population varieties. The one variety is constitutive of different plant genotypes that are closely genetically related. And we investigated the consequences of this huge cultivated diversity on the population structure and evolution of the blast disease, so Pyricularia oryzae on rice. So the first question was, what is the consequence of the co-culture of these the two different rice types, Japonica and Indica? So for that, we sampled 200 isolates, and we genotyped them with microsatellites. And we clearly showed that the population was divided into two genetic clusters, one containing almost all isolates coming from Japonica, and the other one containing isolates coming from Indica rices. And in fact, when we sampled on the field, we see that on Indica rices, the colors are the reverse, so sorry for the mistake, but on Indica plants, we have mostly isolates coming from the Indica-borne genetic cluster, and the reverse on Japonica plants. So here we have a clear signal of local adaptation of the blast population to the two rice subspecies. And then we addressed what is happening within the Indica compartment. So for that, we used paired samples. So we sampled accession on Indica land races, and then we isolated one blast strain per accession, and we compared the genetic structure of the plant and of the pathogen. So on the plant side, we can see that the population is structured according to the rice land races, so represented here by the different colors. So each color corresponds to plants that comes from the same land race. And on the pathogen side, we also have a huge diversity. So we have also 5 genetic clusters, but the genetic structure does not mirror the genetic structure of the plant at all. It means that if you choose, for example, isolates from this cluster, you can find them on all the land races that you have. So there is no genetic cross-structure of the plant and of the pathogen. And then we performed pairwise inoculation. So we inoculated one plant with its own pathogen and with all the other pathogens. And so in this interaction matrix, the redder the color, the stronger the interaction. And what we observed here is that, in fact, all the interactions are compatible. It means that the whole population has circumventing the resistant genes that are present in the indica land races. But the whole matrix is not so red, meaning that the aggressiveness, global aggressiveness of the population is moderate. And there is no signal of local adaptation of the blast population to the different rice land races. So here we have a kind of maladaptation of the global blast population to the different rice land races. So to conclude, as regard the co-culture of Japonica and Indica land races, so we have local adaptation of the blast population, and in fact this is due to the very contrasted immune systems of the 2 rice types, and this leads to, this is equivalent, almost equivalent for the pathogen to intercropping, so facing different plant species. And on the reverse, within the indica compartment, there is no local adaptation of the blast population to the different indica land races. And our hypothesis is that, in fact, this is the signal of the disruption of directional selection and the establishment of a kind of red queen evolutionary dynamics, and that's what we want to verify using population genomics in the future. So to conclude this talk, sorry for the...
Briefly.
Yes, so our take-home message are that in both our case study, we were able to highlight these different mechanisms, and our idea is now in the project that in which we are collaborating, is to use an adequate modeling framework that allows to see how these different mechanisms can be related to each other. And for that, we have the model developed by our colleagues in Avignon, which is called LANCEPI, and which allows to simulate the spread and evolution of a pathogen in different kind of designed landscapes. So I will end it here.
Thank you.
Thank you for your attention.
Yes, and we thank a lot of people that are present on the slide and in the room for certain of them.
So we have time for a very short and few questions, please, in the room. No questions. So there's one up here.
My question is to Elisabeth. As you are presenting about the rice pathogens, so did you observe any agronomic practice effect on this blast or any other local agronomic adaptations on these pathogenic infestations, or these are the global might be effects?
Oh, your question is whether there are differences in agronomic practices? Is that
yeah, the effect of agronomic practices are there or not?
In fact, there is no variation in agronomic practices in the area at least in the area we studied. So everyone is cultivated the rice in the same way. So we cannot answer this question.
Okay, we can take one more small question. Don't sneak in 2 questions. Okay, so there's one question for Fred. so which I hardly understand. Septoria is a dimorphic pathogen. Do you think that the blastospore has a role also in disease epidemics?
So blastospores are present in controlled condition, I think. So it's the equivalent in the epidemic is a pycnidiospore, is the asexual form. Okay. So I don't know about blastospore, probably not, but I'm not sure.
Okay. Okay. So thank you again, and we are going to move to the next speaker.
So our next speaker is Laura MATHIEU, and she's coming from Montpellier, and she's interested in plant-plant interaction. She's beginning her 3rd year of PhD in the lab.
So good morning. Thank you for introducing me and also giving me the opportunity to present today. So, you hear
Can
me?
you speak louder? Yeah.
Ah, okay. So what about processes that occur in mixture due to plant-plant interaction? So today I'm going to speak about plant-plant interaction that modulate the susceptibility to Septoria. So plant susceptibility can be modified in mixture due to epidemiological processes, but also due to plant-plant interactions. And so in this study of Montazo et al., they shown that the cultivar mixtures of durum wheat are less susceptible to the Septoria tritici blotch compared to the pure condition in blue. But when they do a genome-wide association study, they identify a locus at the chromosome 6b where the allelic diversity, so for example for the focal plant which have the allele AA and the neighboring plant which have the allele BB, so this allelic diversity is positively correlated with the STB severity. And so these changes of susceptibility is of the same order of magnitude as the changes due to epidemiological processes. So this shows the importance of plant-plant interactions in mixtures. And when disease susceptibility is modified by plant-plant interactions and in absence of epidemiological processes, this phenomenon is called NMS, so neighbor modulated susceptibility. So to study the neighbor modulated susceptibility, we use experimental design where there is no epidemic to reveal the plant-plant interactions. And so in this system, we observe the phenotype of the focal plants and compare this phenotype between the pure condition and the mixture condition. So we only inoculate the focal plant, and then we observe the phenotype on this focal plant at 17 days after inoculation, so after one infection cycle. And we use a script using deep learning models that we developed at the beginning of my PhD to phenotype this difficult disease. And so as a study model, we use mixture of durum wheat and a strain of Septoria. So for example, this mixture of the focal plant culture cultivated with the neighboring plant Atudur is very interesting because the focal plant culture is more susceptible in the mixture compared to the pure condition. So to better understand this phenomenon of neighbor-modulated susceptibility, there are many questions which remain to be elucidated, but during my thesis and this presentation, we will focus on two questions. So what are the molecular responses of plants to their neighbors, and which genes are required for plant-plant interactions? So for the first questions, we focused on the molecular responses of the focal plant culture, cultivated either in pure condition, so with Atudur, or in pure condition, so yeah, either in mixture with atudure or in pure conditions, so with culture. And so for that, we analyze the transcriptional profile of the focal plant culture, so between the mixture and the pure, using 96 marker genes of different processes that we identify in the bibliography and then optimized. And so to analyze the response to neighbors in kinetic, we collect samples from 4 different time points, so 10 days after sowings, 0 day after inoculation, 7 days after inoculation during the biotrophic phase, and 14 days after inoculation during the necrotrophic phase of the Septoria. So for 0 day after inoculation, there are few genes that are differentially expressed between the mixture and the pure, and among them, most of them are downregulated, so 11 out of the 15 genes. So before the inoculation, we barely observe a response to neighbors in the focal plant culture. But at 7 days after inoculation, the response is much more clear. We observe a massive downregulation of the genes, and this is a general response because we have a significant decrease in the gene expression in the mixture compared to the pure. So at 28 days after sowing, we have a massive downregulation, but also we have another pattern. So here, by analyzing only the defense genes at 7 days after inoculation, so we actually observe the difference between the pure in blue and the mixture in red, but we also observe a different response to the inoculation between the pure and the mixture. So for the pure, the inoculated and non-inoculated sample have different response, but there is no difference for the mixtures. Indeed, the inoculation do not induce the defense genes in the mixture culture atudure compared to the pure condition. So for the second question I would like to answer, so which genes are involved in wheat-wheat interactions that modulate susceptibility to Septoria in focal plants? So we would like to use joint Giraud approaches, so similar to studies on plant pathogen interactions where they try to identify an interaction between virulence genes on pathogen sides and resistance genes on plant sides. So here we would like to do this analysis, but on plant-plant interactions to identify interactions between a signal sent by neighbors and perceived by a receptor in the focals. So the problem of this analysis is that it takes lots of time and of computing power, so we have to optimize the script. And the first strategy was to reduce the number of SNP by selecting SNP from the focals and from the neighbors, which have effect on the phenotype of the focal plants. And so these are preliminary results, but with this first strategy, we confirm that we can detect loci involved in the intergenomic epistasy for the surface of pycnidia in the focal plants. So on the left, you have a heat map of the p-value for all the epistatic combination tested. And so for one interaction tested, it requires 1 second. So if I would like to test all the interaction for all the SNP, it would require 60 years. So I have to optimize the script for sure. And on the right, you have an example of significant epistatic interactions between the focal SNP 008 and the neighbor SNP 606. So in this interaction, the allelic combination of 0 for the focal plant and 0 for the neighboring plant increase the surface of Mycnidia compared to all the other combination. So to conclude, we better understand the phenomenon of neighbor-modulated susceptibility and the response of focal plant to their neighbors with the mixture culture attouture. So in this case, the focal plant culture is more susceptible to the Septoria, and this is associated to a downregulation of gene involved in different processes, but also to a delay in the induction of defense genes in response to the inoculation. So to go further, we would like to identify the signals that triggers modulation of susceptibility. And for the genetics, so we confirm that we detect a loci involved in intergenomic epistasis, and so I have to continue the analysis to optimize the script and then to identify the genes and the processes involved. So I would like to thank the MOMI and G2POP teams that helped me for this work, and thank you for your attention.
Thank you, Laura. Any question in the room? Marc-André? No, no. Ah, do you have a question? Ah, okay. I have one question for Laura. Are there cases where you observe reverse results? That is, cases where the strong of the sorry, where you need to have
with more resistant mixtures.
More resistance, yes.
Yeah, we have these cases in the pathosystem of wheat and Septoria. We have like almost the same number of mixtures that are more resistant and more susceptible, but there are other pathosystems where it's clear that it's more resistant, for example, with the rust.
Yeah, hi. So as you mentioned that some genes are getting downregulated, let me give you an example. There is one gene that calls cellulose synthase, and these genes specifically gets downregulated or sometimes upregulated depending on different pathogenic exposure or abiotic or biotic stresses. So my curiosity is to know whether these genes that are actually shows the internal structure of a plant or an internal anatomical structure, whether these genes are also getting downregulated when these two plants are co-cultured. So did you observe any ultrastructure alterations or anatomical changes during this co-culture?
I mean, the structure in the plant, like in the leaves.
Inside plant, like xylem or phloem or other vascular structures.
For now, we didn't check that, so we don't know. But we have some genes that are linked to the cell state, and they are downregulated. So, but yeah, we didn't check really the morphology of leaves. Yeah, but the genes send some information there. Thank you.
There is no other question. Thank you, Laura.
Thank you.
So the next speaker is Alexey MIKABERIDZE. He's an interdisciplinary researcher in plant health that combines experiment with mathematical modeling and machine learning to investigate plant pathogen interaction. So he's currently lecturer in crop protection at the University of Reading in the UK. And before, he has been a research group leader in plant disease epidemiology at the ETH of Zurich in Switzerland. So please, Alexey.
Thank you very much, Frédéric, and also thanks to all the organizers for inviting me here. I'm very happy to be here and to share with you some of our outcomes on how we can achieve smarter cultivar mixtures to improve disease management. So we are trying to control this disease, Septoria tritici blotch, that is caused by the fungal pathogen Septoria tritici. So previous speakers have introduced the system very nicely. So here you can see the typical symptoms of Septoria tritici blotch, those necrotic, yellow to brown necrotic lesions with pycnidia within lesions. Well, before I go on, I would like to thank all the people involved in this research. So I think this was a very fruitful collaboration between the team at the ETH and the Tunisian teams led by Sarah Ben Barak, who has conducted very nice field experiment on cultivar mixtures in Tunisia. And I'm grateful to all the sources of funding that made this research possible. One of the biggest challenges when we're trying to control Septoria tritici in wheat is that the pathogen has extremely large and diverse populations. So we have recently estimated that in a single hectare of wheat, even in a moderate epidemic year, the pathogen produces more than 5 trillion pycnidia spores. And these are, so this is an incredibly large number if you think about it, a lot of zeros. And these are produced by about almost 8 million unique genotypes, which all inhabit this single hectare of wheat. And so indeed, this pathogen has incredibly large and diverse populations, and this makes it difficult to control, because when we try to apply fungicides, when we plant disease-resistant cultivars, pathogen populations evolve and adapt to control measures. So fungicide resistance emerges and virulent strains emerge in the pathogen population. And so to counteract that, we really need to rethink the way we apply these control measures. So in the spirit of the IPM, Integrated Pest and Disease Management, not only do we need to combine fungicides and disease-resistant cultivars, but we also need to introduce more genetic diversity into our crops. And this is the focus of my talk today, and the focus of the entire session, of course, here. But we need to kind of combine these, not just at random. We need to optimize these combinations in space and over time. And for this, we need to conduct field experiments, but also boost them with novel phenotyping techniques, and also use mathematical modeling so that we have a quantitative prediction of what can happen in the field. And so today, I will talk about cultivar mixtures and show you how we can combine these approaches. And so when we do that, we can actually impede the pathogen adaptation and achieve evolution-smart crop protection, which would lead to more sustainable crop systems. So let's start with field experiments. So this experiment was conducted on durum wheat in Tunisia. The team led by Sarah Ben Barak. And so it was conducted here in the BIOGER field experimental station in Tunisia over 2 consecutive years and 3 replicates. And we used this new phenotyping technique here, where we could collect large numbers of diseased leaves in the field and then use the computer program that identified the boundaries of lesions and also counted numbers of pycnidia within lesions. So we had a very comprehensive measures of disease severity in this experiment. And so we used 3 different cultivars, Karim as a susceptible cultivar and 2 resistant cultivars, Salim and Monastir. And so one of the key questions that we asked here is how does disease severity depend on the proportion of the 2 cultivars in the mixture? So disease severity here is on the y-axis expressed as PLACL, the percentage of the leaf area covered by lesions. And on the x-axis is the proportion of the 2 cultivars. Here it is Karim susceptible mixed with Salim resistant. Clearly, for the pure stand of susceptible cultivar, the disease severity is higher than the pure stand of the resistant cultivar. But what is interesting is what is happening in between for the intermediate mixing proportions. So it turns out that adding just 25% of the resistant cultivar to the susceptible pure stand suppresses the disease severity down to the level of the resistant pure stand and even a bit lower. And I think this is very good news for the durability of such a mixture, because if you have just 25% of the resistant cultivar as compared to the 100% of the resistant cultivar, this will impose much less selection on the pathogen population to adapt to such a mixture. And so we can expect that if we do grow wheat in such a mixture, it will become, this mixture will be much more durable, and so it will the efficacy of the resistant cultivar will be preserved for longer. But what is interesting is that this effect is cultivar specific. When we mixed Karim, the susceptible culture, with a different resistant culture, Monastir, we observed something different. So we saw a rather gradual decrease in disease severity as we increased the proportion of the resistant culture in the mixture. Now, why? Why is that the case? So we thought about it for a long time, but then we realized that we could actually use the modeling, the theory that we developed previously to describe disease suppression in cultivar mixtures. And this theory is based on the modeling approach, on the susceptible infected type of modeling. And so just to introduce you the simplest variant of this model. So this is a compartmental modeling approach where in the simplest case you have just two compartments. H is the leaf area index of susceptible hosts, and I is the leaf area index of infected hosts. And the different arrows represent the different processes. So we have host growth here, important process of infection that converts susceptible hosts to infected hosts. So the rate is the beta times I times H. So it is proportional to how many infected hosts we already have, because these serve as sources of infectious spores. But it is also proportional to the number of susceptible hosts, because if there were no susceptible hosts, there would be nothing to infect. And the coefficient here is beta, is the transmission rate. And of course, infected hosts do not stay infectious forever. They lose their infectiousness with a certain rate, mu times I. Now, I would like to emphasize the transmission rate, because from the perspective of the pathogen, the transmission rate indicates how fast this conversion takes place from H to I. But from the perspective of the host, some host cultivars can suppress transmission rate. And so it serves as a quantification of the degree of resistance of the host cultivars. Now, of course, this simple model has a number of assumptions. It is spatially implicit. We didn't have stochastic processes. These are fairly standard assumptions for this type of modeling. But also, we didn't have latent period, which is an approximation that works surprisingly well, unless your research question addresses specifically latent period. And we also considered endemic equilibrium. So this is not an inherent assumption of the model, but rather we did it just for simplicity. So essentially, after initial dynamic phase, the two-state variables in the model reach a certain stable static equilibrium. And the assumption here is that this is achieved before the end of the season, or at least approached before the end of the season. Now, of course, to model cultivar mixtures, we need at least two different host compartments. So these are now represented by H1 and H2, and these two can have different transmission rates. So, and one of them can be more resistant than the other, and have smaller transmission rate. Now, what do we do with the model? So the underlying model consists of the system of ordinary differential equations. We can solve them and find out how H1, H2, and I depend on time. And then we can compute disease severity. And we can kind of look at it towards the end of the growing season when the endemic equilibrium is reached. And this is quite useful because then we can compare it with disease severity that we can measure in the experiment. But we can also look in the model, how does disease severity depend on the proportion of the two cultures in the mixture? So for this simple model, when we have just a uniform pathogen population with only one infected compartment, disease severity decreases linearly when we increase the proportion of the resistant culture in the mixture. So it's a sort of a boring outcome. Actually, mixture doesn't have, it doesn't make sense to plant a mixture because in order to suppress the disease as much as you want, you are better off planting just the pure stand of the resistant cultivar. But now we can have a more interesting scenario if we consider that the pathogen population can consist of two distinct subpopulations that can specialize on each of the host cultivars. And here we have, of course, it depends on the associated transmission rates. So here we have 4 different possible pathways of transmissions, and so 4 different transmission rates, which we can arrange into this transmission matrix. And the condition for cultivar specialization is that the diagonal elements of this matrix are higher than the non-diagonal elements. And this simply represents the fact that transmission is higher on the preferred cultivar for a particular pathogen subpopulation. And so in this case, when we have cultivar specialization, we have a more interesting non-trivial outcome. So disease severity can be suppressed across the range of intermediate mixing proportions to a level that is lower than each of the pure stands. And you can see, by comparing these two curves, you can see that mixtures indeed can have a substantial advantage in the presence of cultivar specialization. And now we can go back to our experimental outcomes and try to understand them in light of this theory. So we can actually try to fit the two different models to this data. So firstly, we try to fit this model with just uniform pathogen population. And we see, okay, this linear decrease doesn't really explain the patterns we see in the data. Now we try to fit a somewhat more complex model that takes into account the possibility of cultivar specialization. And as we can see, it actually does capture this key pattern of suppression of disease severity at intermediate proportions. And this visual impression is also confirmed by the model selection with archaic information criterion, where it is substantially lower for the blue curve, the model associated with the blue curve. So this is our preferred model in this case. Now, let's look at the different mixture of Karim Monastir. So Karim susceptible with a different resistant cultivar Monastir, and in this case, both models, when we try to fit it, both models converge to this simple case of a uniform pathogen population where we observe simply a uniform, simply a linear decrease in disease severity as we increase the proportion of the resistant cultivar in the mixture. So this is our preferred model in this case. Right, so one of the graphs went missing, so it's this one. So, so the conclusion is essentially that cultivar mixtures have an advantage when pathogen population exhibits cultivar specialization. And so our kind of this model, the comparison between the model and the experimental outcomes allows us to hypothesize that in this case of the Karim-Salim mixture, the the reason why we see this pattern of decreased severity and at intermediate mixing proportions is that the pathogen population exhibits cultivar specialization. So we can test this hypothesis. But in the case of Karim-Monastir mixture, the kind of explanation for this pattern would be that the pathogen population does not exhibit cultivar specialization. And that's why there is no preferred intermediate mixing proportion here. And this can be, so this is still a hypothesis, because there can be a number of alternative explanations of these patterns in the data. But this model experiment comparison leads to a number of testable predictions that we can test in the future experiments. So firstly, as I mentioned, pathogen population, we predict that pathogen population exhibits cultivar specialization for one of the mixtures, Karim-Salim mixture, but not for Karim-Monastir mixture. And we can look at the pathogen population and indeed test this hypothesis in the future. And secondly, with cultivar specialization, pathogen population is predicted to be more diverse across intermediate mixing proportions, and we can again test it. So just sample the pathogen population across, in this case, across the intermediate mixing proportions. You might remember the blue curve. I think we have a fantastic audience here, and I will be very happy to take any questions. Thank you.
So thank you very much, Alexey. We can take some questions.
Hi, Alexey. Very interesting talk. Thank you. I'm over here.
Oh, okay.
Hello. I just wanted to ask about your assumption of the endemic equilibrium. So I guess if you're assuming, that the pathogen reaches its equilibrium, you could miss out on the, differences to do with kind of rate of spread and that kind of thing. I'm wondering how you think relaxing that assumption could potentially affect your results, or whether it matters at all.
So I think this can matter in the initial stage, but I think as long as you at least approach the endemic equilibrium, so you're kind of maybe halfway through to the end of the season, this should work. But I think it's an interesting question about the dynamics. The initial dynamics can be different depending on the initial conditions, depending on the inoculum from the previous year. So I think this is something to explore.
Thank you.
Well, thanks.
Yeah, I have one question. What do you mean by specialization? It's virulence or aggressiveness with a fitness cost, for instance, or both of them?
So in this case, it can be a combination of both, because the only thing that's required is that the associated transmission rate should be higher. So the diagonal transmission rates should be higher than the non-diagonal ones. So, but the underlying mechanism can be either quantitative resistance or qualitative resistance. It can be anything.
And can you vary the strengths of this specialization in your model?
Yes, absolutely. Yeah, that's what we did when we fitted the model to the data. Yeah.
Alexey, thanks for the talk. I was just wondering whether on the CAREM-SAREM mixture, the experimental data you showed, if I look at the actual points where you have the 75-25 and 25-75, they seem to be just a tad lower than the 50-50. So it seems that maybe there is an extra mechanism besides cultivar specialization that might explain also why you get a sort of a little peak at 50-50 rather than 25-75.
Thanks. I think this is an interesting observation. I think with this sample size, we could not distinguish between these two, but I think if we try to increase the sample size and kind of try to look in more detail, what do you think could be the mechanism? Okay. Mm-hmm. Right. Yes. Yes. Thanks for that. Yeah.
Yeah. Thanks, Alex. As a follow-up, so what was the sowing pattern of this in this experiment?
Oh, so it is an important fact that I forgot to mention. So it's just uniform, spatially uniform. So the seeds were mixed in a uniform manner.
Ah, okay. Because that could maybe an explanation if you would have done row sowing or strip sowing. But
right. No. Yeah.
There's a question on the app, which is for you, Alexey, but could also be for Laura. The question by Ashok Kumar is, how does the cultivar mixture affect monocyclic pathogens?
Oh, that's a good question. So here we focused on the polycyclic epidemic, but I think we would need to better understand what's happening between the seasons, and I think Frédéric has a better idea about that.
Oh, there's the last question in the room.
In this case, we have positive cultivar specialization. But do you think that it exists also negative cultivar specialization, that we have this case? We have the resistant, the susceptible, the resistant, but the cultivar mixture are more susceptible than the susceptible variety itself. Do you think that it is possible also?
I think it's possible, and I remember seeing such results, but we didn't observe it in this experiment.
It is possible, and I see this case in Tunisian variety also.
Oh, okay. Oh, it's interesting.
In past studies.
Okay.
Okay, so if there is no further question, I would like to thank again Alexey. And call for our last speaker of this morning. So this is Tiphaine VIDAL. So Tiphaine is a plant disease epidemiologist at INRAE, Saclay, northern France, as you know. She worked in the past on the architecture of wheat mixtures and the mechanisms that would favor disease control based on architecture. She's now also interested in epidemics of leaf rust on wheat. But as far as I understand, today she's going to tell us more about, again, our favorite system, which is wheat and Septoria disease and In particular, she's going to tell us about the impact of local conditions on the functioning of those mixtures. Hopefully she can make it.
C'est pas la bonne présentation.
Ah. Je sais pas. Ils ont entendu ou pas, selon toi? Ouais. Il faut aller à la caisse. Non, c'est la dernière. Ouais, ouais, ouais. Ah. Ah. Yeah, ouais, ouais. Je peux faire. So while we wait for the problem to be fixed, I would like to deliver a quick conclusion, let's say, of this session. And although the session is not over, oh yeah, sorry. So although the session is not over, I hope you enjoyed this session, and I think that based on the dynamics of the question, that many of you are quite interested in this topic, and I'm very glad to see that, because we do think as the organizers that we do think that intra and interspecific mixtures are really key agronomic tools that needs to be much more deployed, and there were questions on that actually on the app. They need to be deployed more than they are actually. So, and just to give you a hint, because there was a question in the app, why isn't there more diversity actually grown since we see those strong phenotypes? And just in France, be aware that if you look at the field, wheat field, okay, actually currently 12% of the fields are being wheat mixtures. Okay, when we sample, when we ask the farmers, we discovered that it's not just homogeneous, it's actually already 12% of the surface of wheat in France, which is covered by mixtures, okay? So that, and it's increasing, it's an increasing trend. And I was very pleased actually to hear Annalisa mentioning that mixtures, they also have a beneficial effect on carbon sequestration. And I think this is very important to further study, okay, the impact of those mixtures on carbon fixation, because as you know, I mean, diversity and no pesticide is one challenge for us, but carbon fixation is a challenge for all of us, okay? And so I think these systems, they also have this extremely nice property to fix carbon better than monoculture. And keep that in mind, okay? Okay, so do we have the presentation or not? So, no?
Not yet.
Okay, so I keep going. All right, so, okay, so I do the, no. So Tiphaine, so we wait for Tiphaine. Okay, so I hope I'll come back later to highlight a few points I found quite interesting to highlight to this community and to prepare the future of this research. So we need the speaker now. Let me see, it's wheat, Septoria disease. Okay, so please welcome Tiphaine. Thank you.
So hello everyone, thanks for your patience. At last, my presentation. So today I'm going to talk about the same case study as previous speaker, which is Septoria tritici blotch on wheat cultivar mixtures. So the good news, I will be able to skip part of my context because it had been presented before, and I'm going to focus on factors that impact the mixture effect locally and focusing on different European sites. So I mostly skip that. So you know that there can be a mixture effect on Septoria tritici blotch. So I think we've seen that before. One important criteria for mixture design is the contrast of resistance between the mixed cultivars. But you might know that the resistant level for a determined cultivar can vary with local conditions. For example, if you have a different composition of pathogen population, if a particular gene is broken down locally, you will have a different resistance, and you can also have other environmental effects. So the objective of this work was to assess the disease control provided by a cultivar mixture in different locations in Europe, and also to study how this mixture effect was related with other factors, including the local resistant levels of cultivars in pure stand, but also some climatic factors. So for that, we work with two cultivar mixtures, so with two cultivars. So Apache, which was mentioned before, it's a susceptible cultivar which has been grown for quite a long time in France. So we suppose that our ZIMO population are adapted to this cultivar. And the other one is Cellule, which is a more recently introduced cultivar carrying a major gene. But this gene was broken down relatively quickly. And we know that its resistance is still effective in some places in Europe. So in the field, we were able to distinguish these two cultivars because one has a beard and the other hasn't. So this is really interesting because I will show you later the disease level of the different cultivars within mixtures, and that we will be able to compare with the pure stand. So we studied the variation of mixture effect in local conditions. So we had 7 sites in different European countries, as you can see on the map. And we carried out the experiment for 3 years. And in each place, we had Apache and Cellule in pure stand and the mixture in equiproportions with 3 to 4 repetitions per treatment. So what we measured basically is disease severity. So if you remember the Septoria tritici symptoms, we measured the area covered with pycnidia, which are these tiny dots, black dots. And we also computed incidence. And we carried out this disease scoring by leaf layer. And for the rest of my presentation, leaf 1 will be the flag leaf, which is the top leaf. And then leaf 2 is lower, and we get lower. So we measured 15 plant per cultivar and per plot, and we carried out statistical analysis with R. So one first thing we observed, it was quite expected, but we were able to check that, is that cultivars definitely behave differently across trials. So here, each point on the graph corresponds to one leaf layer. So it's a mean of 15 leaves, in fact, but one leaf layer measured in one site and 1 year. So here you have the disease incidence on Apache compared to the disease resistance, disease incidence on Cellule. And basically, you have the same representation for severity. And how can we read this? Here we have the case when Cellule is resistant, which means that we have high disease pressure represented with high severity on Apache. But despite that, Cellule is resistant. So that's the expected case when the resistant gene is efficient. But we have also other cases. And relatively surprisingly, we have places and dates when Cellule is as susceptible as Apache. So it has completely lost the efficiency of its resistance. And as you can see, there are many intermediate cases. So we call this difference between Cellule and Apache the resistant contrast. And we will see later how it was related with the mixture effects we observed. So starting with Apache, here we compare the absolute mixture effect. So basically, we take the disease severity in pure stand, and we compare it to the disease severity in mixture. And basically, if you are here, it means that we reduce a lot the disease severity in mixture. And we compare that to the contrast between cultivars. So here, if you are on the right, it means that Apache is much more attacked than Cellule. So here we have a positive correlation, which is also significant. And we see that we have high mixture effects when the resistance of Cellule is quite effective. So that's really the classical case we are looking for when we design mixtures. It's to protect the susceptible cultivar with an efficient resistant source. And we also have the case when Cellule is susceptible and in this case, basically, there is not much effect. Sometimes we have a little bit more disease on Apache, but it's not significant. So let's now look at cellule. Here we see also a relation between the contrast and the absolute effect, mixture effect on cellule, but it's negative. So how can we read that? When cellule is resistant, in rare cases we have slightly more disease on cellule in mixture, but once again it's not significant. In most cases we have low effects of mixture of cellule, but interestingly, in some specific cases, cellule can be protected as Apache when it's susceptible, as if it was a susceptible cultivar. So, so here I gave you the results for one factor that we studied, which was the resistant contrast, and we see that it impacted the mixture effect for both Apache and cellule. So that is consistent with the literature. For example, with modeling studies suggesting that more contrast improves the protection of the susceptible cultivar. But we also studied other factors. For example, disease pressure, which we estimated, we used as an indicator Apache in person, as Apache is susceptible. So here, this had an impact only on the mixture effect for Apache, but not for cellule. So this is also consistent with the literature, which shows the tendency that with more disease pressure, we tend to have higher mixture effect. And finally, we also used a very basic indicator, which was mean temperature from sowing until the scoring date to represent the number of cycles. And we saw that this was significantly related with mixture effect for cellule only. So this also could relate to literature results, for example, indicating that with more cycles, the mixtures tend to have more effect. So now we have investigated the basic variables, but we could also get more into details, for example, using epidemiological indicators, for example, the number of days adequate for infection using an infection model, or the number of latency period, or maybe the rain events that are adequate for dispersal. We might also test a relation with local frequency of STB16 virulence, but for that we will need molecular tools that are not available at the moment. And at a perspective, it would be interesting to study the factors that impact the contrast between cultivars, and we could study that in particular using varietal assessment network data, and also the probable relation of mixture effects with pathogen population structure. So that could be investigating using either molecular tools, but also modeling approaches. So in conclusion, this result suggests that we see that the mixture effects vary locally, and maybe if we know better the mechanisms that impact this variation, we can adapt the mixture design to local conditions, and this could contribute to better disease protection and resistance durability provided by cultivar mixtures. So thank you for your attention. And if you have questions,
thank you, Tiphaine. Do we have questions in the room? Yes, here, left, over there.
Yeah, thank you for your talk and also your thoughts. One question, so the gist of your talk was how a susceptible cultivar could somehow be protected to a certain proportion of a resistant cultivar, but also vice versa. So did you also look at different effects, so how, let's say, the resistance of the sessile cultivar could also be somehow prolonged or protected through the susceptible cultivar?
Yeah, so that's really what we saw here. That basically we can consider that cellule is a resistant cultivar because it possesses a major gene. But in fact, when the situations when we have a significant effect is when it's relatively highly attacked. So when it's resistant, we don't have significant effects, also because the level of disease is quite low already. So to have an effect, I suppose you must have some disease if you have very low disease. people sometimes are afraid that you might have more disease on the resistant cultivar mixtures. Up to now, we have no significant increase in disease on resistant cultivars. But of course, if it has a low disease level, we don't have a mixture effect.
I have one question here. Tiphaine, here? So to me, it's always intriguing to see that the more disease, the more effect we get finally. Okay. I mean, it's not intriguing, but what is your idea behind the mechanism behind? In particular, I'm thinking about induced systemic resistance, where a spore from a susceptible plant is landing onto a moderately susceptible resistant, thus inducing some protection which will be efficient against another spore and so on. So do you know if induced susceptible resistance has been quantitatively speaking measured in cultivar mixtures? And if you don't, does somebody know in the room if ISR has been measured in the field and quantified?
So I think there are quite relatively ancient work on induced resistance. It's quite difficult to show in field condition. It's really hard work.
It's awful.
Yes, but in general, when we are in field conditions, so I think it's relatively intuitive that we get more effects when we have more disease. Because when we have more cycles, at each cycle you have the possibility to have different mechanisms: the barrier effect, the density effect, maybe impacts on the genetic structure of the population. So all these impacts, they can occur during the different cycles. If you have no cycles, nothing will happen. But after some time, if you have too much disease, then the system can
it's plateauing.
Yes, it can reverse a little bit. But most of the time, in field conditions, it's quite hard to distinguish the effects because they are all interconnected.
Yes, of course. Yeah. Okay. Are there any further questions to Tiphaine? No. So again, thank you, Tiphaine. So before we close this session, indeed, I would like to highlight a few things about this topic, which to me is again, I would like to insist on it. This is a very interesting object to be studied by biologists or by geneticists, epidemiologists, modelists, and so on, or even geophysicists. And so I do encourage you to speak positively about those systems, because they are known by some of us, but mostly unknown. And they have so nice and so massive properties and positive properties for agriculture and obviously for the environment. But what we see through those talks is that there are still many unknowns, okay? Many things we don't really measure, we don't know. There are many unexpected observations, many paradoxical observations, like there's no dilution effect sometimes, there is no effect of richness, which to me is quite surprising, because this is somehow making the BEF hypothesis from ecology non-valid. And so, again, there are many paradoxical observation, and as scientists, that's probably what you're looking for, things that you don't understand, okay? And there are many things here to be understood yet, so I encourage all of you, including young people, to invest your time and energy onto studying those systems. And of course, there are new players in the systems like microbiome. Microbiome is entering this field. It was more or less absent from this field a couple of years ago, but now it's in, and obviously the interactions in the soil between the plants is going to modify exudation, which is going to modify microbiome biology, which is going to modify plant-soil feedback, and so on and so on. So clearly, those systems, they have very many what we call emerging properties, okay, that we can hardly predict from studying each isolated organism. So they need to be studied as a group to better understand. We saw that there were some also new levels of regulations that were unsuspected so far, like the plant-plant interaction level, which when we heard about the effect of, I think Bob mentioned, to me it sounded like there are actually plant-plant interactions in your search that you are detecting, or the fact that the 5 species that Annalisa has detected as being very positive in decreasing disease. If you look at those species, they are all Fabaceae, which are famous to produce some molecules, exudate molecules, which are very famous to be signaling molecules. So is it not just that we are looking at, okay? Not diversity, but just physiological, basic physiology between 2 organisms exchanging signals, okay? So this is raising many, many questions, and again, as scientists, we should be all excited about the unknowns, because this is what justifies our work. So, because we think this topic is very important, I encourage you to, again, to talk about these systems, and we think this community, or at least the people who are interested in working on those very interesting objects, this community requires to be further consolidated at the international level, because we need all your different skills. And there's no society really dedicated to studying those objects, and I think it would be worth if we were organizing some sort of society dedicated to organizing conferences and stuff around those objects, which I believe are critical and key to solve major issues, present and future issues. In that perspective, we are going to organize with some of our Chinese colleagues that are present here from Yunnan, who are very much into agrobiodiversity as a major key lever for agriculture. So we are going to organize next year a satellite conference dedicated to inter- and intra-specific diversification for crop protection. So there's no yet a date, but I invite you to stay tuned on that topic, because we will have the capacity to invite many speakers from all over the place, and to try to start or initiate a community on those objects. So again, please stay tuned on that topic. There's no information yet, but there will be soon. And if you're interested in studying those systems, in particular in southern countries, we heard that Bob said, and that's very true, that Africa and Asia are key leading continents on those topics, because they do practice those systems. Okay, they have a huge experience on those systems. So it's clearly an international issue and object. And I hope we'll see some of you soon, soon, next year in China, further discussing of what we know, what we don't know, what's surprising and what is not surprising. So with this, I want to thank again all the speakers of this session and your contribution to the discussion. Thank you very much.